Thus far in Chapter 9, we have discussed the idea of gene expression, recombinant DNA technology, genetically modified foods, and lastly, we're talking about genetically modified humans. And we've already discussed stem cells. What I'd like to cover with you now is what exactly is the Human Genome Project, and then lastly, we'll cover gene therapy. So in terms of where we're going to head, we'll initially talk about what were some of the early human genome maps. We'll then talk about how do we actually sequence the human genome. We'll then talk about the project itself and what has been learned from this project. We'll also talk about the protein coding regions, which is only 2% of the genome, and we'll finish up talking about what is the rest of the genome. So if we're looking at a chromosome and we want to identify exactly where on that chromosome a particular gene is located, we need a genetic map. Initially, these maps were generated using physical maps, which were based on recombinant DNA studies where they were looking at recombination events that would take place during meiosis. And this would give you an approximation of where genes were located in terms of how far they were from one another. The more accurate way to figure out the location of genes on a chromosome is actually by doing DNA sequencing. The way that these physical maps are made from the sequencing data is that we are basically making a copy of the DNA. So when we talked earlier in the semester about DNA replication, we needed several key components. We needed things like the DNA itself. We needed a primer that would provide a 3' hydroxyl end for the DNA polymerase to attach to so it could begin to polymerize that new strand of DNA. We also needed ribonucleotides, the deoxyribonucleotides such as the A's, T's, the C's, and the G's. By way of reminder, we had talked about DNA polymerase, which is what's binding that incoming nucleotide to the strand of DNA that's already there. And for it to do that, it requires a 3' hydroxyl on that carbon that's on the deoxyribonucleotide. So the question to you is, what would happen if there was no 3' hydroxyl on the deoxyribonucleotide? And the answer is that DNA synthesis would stop. And that's the key to how DNA is sequenced. So when we want to sequence DNA, we actually use a ribonucleotide that does not have a 3' hydroxyl as shown here. These are called dideoxyribonucleotides. And when these are inserted into a strand of DNA that's in the process of being polymerized, the whole sequencing reaction will stop because the DNA polymerase can no longer bind to a 3' hydroxyl group. So if we wanted to do a sequencing reaction, so let's say for instance you wanted to synthesize this strand of DNA, we have a primer that is complementary to the 3' end of this DNA strand. We have DNA polymerase that will polymerize this new strand. We have deoxyribonucleotides, the A's, the T's, the C's, and the G's. And then we've also included some dideoxyribonucleotides that lack that 3' hydroxyl group. And these dideoxyribonucleotides are also radioactively labeled. So when we go to perform the reaction, we have a mixture of the DNA, the primer, the polymerase, and the deoxyribonucleotides. And then we add just one of the 4 dideoxyribonucleotides into a tube. So as shown here, you have your mix plus dideoxy ATP. In tube number 2, you would have your mix plus dideoxy CTP. Tube number 3, you'd have mix plus dideoxy TTP. And lastly, you'd have the mix plus dideoxy GTP. You then allow those sequencing reactions to occur. And what you have is a competition between the deoxyribonucleotides versus the dideoxyribonucleotides. So So if a dideoxyribonucleotide forms that complementary base pair as opposed to the deoxyribonucleotide, then the synthesis of that DNA strand is going to stop. So once you've done these 4 reactions, the next step is to go ahead and denature the double-stranded DNA by heating it up to somewhere around 96 degrees. You're going to then put that into an agarose gel. It's going to separate out based on size, with the smallest fragments moving the farthest down the gel. Since I mentioned that dideoxyribonucleotides are radioactively labeled, you're going to go ahead and expose that to X-ray film, and then the following day you'll be able to read the film and analyze your results. So your results would look something like this. So you had your 4 different dideoxyribonucleotides, and you can systematically work your way up from the bottom of the gel. So as you can see here, this gel, if you're starting at the very bottom, you can see in the lane marked with an A, you have a black band. And then the next lane on the far right with the pink C would be your next ribonucleotide. So if we were going to read this strand of DNA, it would be ACG, AG, AT, 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 and you would systematically work your way up from the bottom of the gel to the top of the gel, and you would be able to figure out what was the actual sequence of that DNA. So here on the left is showing you what a typical sequencing gel would look like, but there's a lot of limitations to this type of method. It's very labor intensive. You're working with a lot of radioactivity. It takes hours just to run your samples through these gels. They're very thin gels, and it takes a long time to separate out those bands. And once you've done that, you can only read somewhere between 200 and 400 nucleotides per reaction. This is also called the chain termination method, which I will let you think about why that might be the case. So if this is still confusing to you, which I would presume it might be, please go to the following website, which shows you what this looks like in an animation that's quite helpful. Okay, so that is the Sanger method, but in the early 90s, there was an automated sequencing technology that was developed. So rather than adding the dideoxyribonucleotides that were radioactively labeled, we could now fluorescently label these tags. So you could do all 4 reactions in one tube because you attached 4 different fluorophores onto the A, the T, the C, and the G of the dideoxy form. You would then run these samples through a capillary tube that was filled with gel. The DNA would separate out based on size, much like it does in the agarose gel, and the DNA sequence is then read by a laser. So here's the fluorescently tagged dideoxyribonucleotide. So you would then generate a set of data that looks like this, that basically has a printout of what the actual sequence is. So rather than being able to read 200 to 400 nucleotides per run, which would take several hours to generate that gel and then an overnight exposure to the film, you could actually now, with this automated sequencing method, read up to 100,000 base pairs per hour. You can set this up to run overnight with lots of samples, and you can get a massive amount of data in a short amount of time. The downside is these machines cost somewhere around $300 for each one. Okay, so that leads us to the Human Genome Project. So the main goal of the Human Genome Project was to map and sequence the entire human genome. It officially began in 1990, and it was set to be complete by 2005. The initial leader of the Human Genome Project was James Watson of Watson and Crick. Also at the time at the NIH, Francis Collins was heading up this project, and R.A. Petrinos at the Department of Energy, and they were both working under James Watson as the head of this project. The approach at the time when they began was that chromosomes were distributed to various labs around the world, and this was considered a worldwide effort, and it was called the International Human Genome Sequencing Consortium. The overall cost was expected to be somewhere around $3 billion. So what were the goals? They wanted to identify all 100,000 genes in the human DNA. We now know that there's far fewer than that, but that was what it was thought of at the time that the project began. They also wanted to make these genes accessible for further study to anyone who wanted to research them. They wanted to sequence all 3 billion base pairs. And this was somehow needed to be stored in an appropriate database. The goal was to use the Sanger method, but hopefully that new technologies would come along as this process began, which I've already described to you, the automated sequencing machines that were developed partway through this project. They also wanted to not only create a database, but create tools for that database so that meaningful analysis could take place. Lastly, they wanted to set up a group to address ethical, legal, and social issues that would possibly arise due to this project. So James Watson was the initial leader of this project, but only for a few years. He actually stepped down in 1992 due to internal conflict. So Watson, who we've already discussed, oftentimes had issues with other people, such as Rosalind Franklin. At the time of the Human Genome Project, he was at odds with a director at the NIH named Bernadine Healy, and especially at odds with Craig Venter, who was also working with Healy. So Watson's desire was to systematically sequence the entire chromosome, and you call this chromosome walking, and just walk your way down the chromosome. Healy and Venter wanted to focus only on something called express sequence tags, and they also felt that the government should patent these genes. Since it was government-funded research, they felt that the government should have a patent and legal rights to the genes that were discovered. And Watson vehemently disagreed with this. So what exactly are express sequence tags? Well, they're the genes that are actually expressed. So if you were to isolate the genes that were expressed in a cell, that would be the RNA. And you can then do something called reverse transcribe that RNA. So you make a copy of it using cDNA, and then you can degrade that RNA copy and create double-stranded DNA. So you are not looking at all of the nucleotides in your chromosomes, but you're just looking at basically the genes that are expressed at any one time in the cell. So Watson and Venter could not agree, and Watson finally gave up and he resigned in April of 1992, and leadership was passed on to Francis Collins in 1993. By the early to mid 1990s, sequencing was having this major transition to using these fluorescently labeled tags. And so using high throughput DNA sequencing greatly accelerated this project. In 1996, those who were funded by the Human Genome Project, since it was government funded, they all agreed to release their data within 24 hours so that anyone in the scientific community could immediately begin to do research on what was found just the day before. So by 1996, a rough map was developed. You can go to the address down below and you can see some of those early genes that were identified. And they had immediately published information on approximately 16,000 genes. So at this point, Venter decides to leave the NIH in 1998 because he thinks that he can do this project much better on his own and without the restrictions of the government, as well as not having to work with the World Consortium that was working on this project. So he begins his own company called Celera with a scientist who had come from Applied Biosystems. Applied Biosystems was the company who developed the high throughput sequencing machines that I mentioned earlier. So here's a picture of Venter that I like of him, half as a scientist, half as a businessman. So his goal was to sequence the entire human genome, not by 2005, but rather by 2001, and rather than it costing billions of dollars, he was going to do this for about $300 million. So basically, when he did this, it began a mad race between the world and Venter at Celera to see who could sequence and complete the Human Genome Project first. So he wanted to use a method called shotgun sequencing, which I will not go into detail on. He also was able to obtain 300 automated sequencing machines that would then be running 24 hours a day around the clock. So Venter was actually able to better his prediction, and he, along with the World Consortium, came up with a working draft that was completed in June of 2000. Actually, Venter had come to the White House in April of 2000 saying that he had completed a working draft, but he held off and waited for the government as well as the world to catch up to him, and they declared that they had completed this working draft in June of 2000. So here we have a picture of Venter on the left with Petrinos in the right at the head of the Department of Energy, and Francis Collins on the right doing this super awkward three-handed handshake. And you have a picture of Venter and Collins on the right smiling, although I'm sure it was quite contentious for many years there. So what they had discovered was sequencing data and the chromosomal locations for the genes, for about 90% of the genes. The final version was ultimately completed in April of 2003. So here we have Venter and Collins on the cover of Time magazine. They also each had the lead article, Collins in Nature and Venter in Science magazine, that came out at the exact same time, showing their discovery of the human genome. And this is in February of 2001. So what has since been sequenced? Well, the mouse gene came out in December of 2002, the chimpanzee, as well as the dog. You can see here the dogs are looking at a picture of Watson and Crick studying the model of DNA. We also have the sea urchin, as well as the possum, and many others as well. So what are the benefits of the Human Genome Project? It is used in everything such as molecular medicine, forensics, bioarchaeology and anthropology, human migration patterns. It's used extensively throughout research. So what did we learn? Well, we know that the human genome contains approximately 3.2 billion base pairs. The average gene is about 300 base pairs in length, although there's some that are drastically shorter and some that are much, much larger. People are 99.9% similar to one another. The male mutation rate is twice that of females. They do have more mutations, but that's also evolutionarily advantageous. So what we've determined is that these genes clump together in what we call gene-rich regions, and these are interspersed with massive desert regions where there's no genes present. On average, the human gene will make 3 different proteins due to splicing, but simpler organisms just make 1 protein per gene. We know that chromosome 1, which is the largest chromosome, has 3,168 genes, and the Y chromosome, which is the smallest of the chromosomes, contains only 344 genes. The other 98% of the genome contains things like repeat sequences, as well as stretches of cytosine and guanine. We call these CG-rich regions. And these CG-rich regions are often adjacent to gene-rich areas. So they're thought to somehow be involved in gene regulation, but we aren't sure. How does all this information affect you and me? Well, you can go to the following website called 23andMe, which I would highly encourage you to do, and you can actually get much of your own genome sequence for as little as $99. A lot of what they do is looking at migration patterns and your history, but you'll also get back a massive amount of information regarding your health. So if you go to the following website, you can learn a lot about genetics in general. So I'd like for you to go to the following website and answer the following 3 questions. What % of your human genome is shared with a chimpanzee? What % of your genome is identical to your colleague or friend? What exactly are SNPs? How are they usually generated? And how many are there? So take a minute and go in and check out that website. So we've talked extensively about nuclear DNA, but is that all the DNA in your cell? And the answer is no. There's actually additional DNA, which is your mitochondrial DNA. Mitochondria, you'll remember, is considered the powerhouse of the cell. This is where cellular respiration takes place. What do we know about mitochondrial DNA? Well, it's a circular DNA containing 37 genes. A lot of the genes are involved in oxidative phosphorylation. We have some other key genes. This is called the Leber hereditary optic neuropathy. There's also other genes involved in cardiomyopathy, as well as this MRF gene, which is the myoclonic epilepsy and ragged red fiber disease. So there are a few disease states that are associated with mutations in mitochondrial DNA. The key here is that your mitochondrial DNA is inherited solely from your mother. When the sperm fertilizes the egg goes on to destroy all the organelles from the male's gametes, except for the nuclear DNA. Each egg has somewhere around 200,000 mitochondria. The male sperm is only contributing its nucleus, its nuclear DNA, not the cytoplasm. So we know that all your mitochondrial DNA is coming solely from your mom. An important area of the mitochondrial DNA is the hypervariable region. This hypervariable region is able to undergo mutations at a rate somewhere around 100 times that of nuclear DNA. So this region is used to track lineages over time because it's an excellent area to see where changes can be made in the DNA. So if you're going to look at a pedigree of a particular mitochondrial disease, what you'll see is that if a female is affected, all of her offspring will be affected, whether they're male or female. However, since only the females are passing on their mitochondrial DNA, only her daughters will then pass on that mitochondrial disease to her offspring. So as you can see here, the daughter on the far left passes on the mitochondrial disease to all of her offspring, whereas the second child, which is a son, none of his offspring are affected because they're obtaining all their mitochondrial DNA from their unaffected mother. In this situation, Mendelian ratios will not be observed. Here's a picture of Tsar Nicholas II of Russia with his wife and 4 daughters and his son. We often discuss the role of hemophilia in the royal families. However, for our discussion now, we're going to highlight the role of mitochondrial DNA in solving a mystery. In 1917, during the Bolshevik Revolution, Tsar Nicholas Romanov II was overthrown, and in 1918, the Tsar and his family were reported to be executed. However, rumors persisted that Anastasia, the youngest, had escaped this execution. And in 1922, a woman came forward claiming to be Anastasia, who was living in Berlin. She later moved to the U.S., and she died in 1984, claiming throughout her life that she was indeed Anastasia, the youngest daughter of Tsar Nicholas II. Here we have the royal family tree, specifically highlighting how hemophilia was passed down through the families. What I'd like for you to look at is Alex Serena, who was also married to Nicholas, and Nicholas is not depicted on here, but Alex Serena's offspring are shown as 3 circles, which indicates 3 daughters, the 4th daughter, who's Anastasia, and then Alexis, who is her son, who did have hemophilia. So the question is this woman who's claiming to be Anna Anderson, is she actually Anastasia or is she not? So knowing that mitochondrial DNA comes from your mother, is there any living relatives alive who we could compare the mitochondrial DNA to who should be a match to Alex Serena's daughter, Anastasia? So take a minute and look at the family tree and see if you can figure this out. Indeed, we can look at Prince Philip, who's married to Queen Elizabeth, and his grandmother, who was Princess Victoria, was the oldest sister of Alex Serena. So indeed, the mitochondrial DNA from Prince Philip should match the mitochondrial DNA of Anastasia. Once the mitochondrial DNA was analyzed, it was proven after her death that indeed Anna Anderson was not Anastasia. Anastasia, in fact, had been killed along with her family during the Bolshevik Revolution. So here's a picture of the 4 sisters. Anastasia is the one on the far right. And here's the woman, Anna Anderson, who claimed for her entire life that she was of the Russian royal descent. How is mitochondrial DNA used to analyze human migration patterns? Well, mitochondrial DNA is very abundant in the cell. A single mitochondria can contain 10 copies of its DNA. Each cell contains hundreds of mitochondria. It's very durable because it's circular, unlike our nuclear DNA, which is in chromosomes. The circular DNA is not easily degraded. In that hypervariable region, it has a high mutation rate, much higher than nuclear DNA, and this allows you to track in a shorter amount of time any changes that are happening, allowing you to really analyze migration patterns. Well, that's true for mitochondrial DNA, which is coming from your mom, but what about the Y chromosome? So we know the Y chromosome can only be passed from father to son. It is nuclear DNA, but it can also be analyzed to look at paternal lineages of populations as well. So using the mitochondrial DNA and the Y chromosomal DNA, you can actually look at migration patterns of your family over tens of thousands of years. You can do this by joining the Genographic project, which is run by National Geographic. You just take a simple cheek swab and you can be a part of this project. Using this type of DNA, they've been able to determine human migration patterns over tens of thousands of years.